Good afternoon, everybody. Today we're going to talk about cell respiration. In this particular Prezi, we refer to it as chemoheterotrophic energy processing. Remember that autotrophs are plants. Auto means self and troph means feeding or food. And that means that they make food within their own bodies. So that's photosynthesis. Autotrophs do photosynthesis and make their own food. Chemoheterotrophs need to eat in order to gain their energy. So we're talking about cell respiration that's happening in organisms that can't do photosynthesis. All right, so let's move on and take a look. Big question, how do living systems process energy is what we're going to be looking at today. A quick recap. First of all, there's a reciprocal relationship between chemoheterotrophic nutrition and photoautotrophic nutrition. The inputs of one are the outputs of the other. Food is what gives us our energy, and this chimpanzee would die if it didn't eat, so we don't want that. Very silly author. Okay, so take a look at the two diagrams. Super important you get the reciprocity here, the reciprocal relationship here. So notice that water and carbon dioxide are going into the chloroplast as well as light from the environment. And then the chloroplast is producing the oxygen and organic molecules like glucose that are then going into the mitochondria. The mitochondria is then producing the carbon dioxide, and it's a cycle that goes around. One of the outputs of the mitochondria is ATP, and that's really what we're going to be focusing on today. We'll talk about photosynthesis later. And then with the release of ATP is always the release of some heat. Some of the energy gets released to do work, and some of the energy gets released as heat. That's actually a bad thing. You don't want a lot of heat to be released, but that's just part of releasing energy. All right, moving on. So the reciprocity or the reciprocal nature of those two, the photosynthesis and the cell respiration, explains this crazy, please take a moment to look at it, that aerobic cellular respiration, aerobic means with oxygen, takes C6H12O6, glucose, and oxygen, and makes or produces carbon dioxide and water. It also makes some ATP that they don't include in this particular equation because they're trying to show you, now look at photosynthesis, look at the products of cell respiration. They have now been moved to be the reactants in photosynthesis. So the carbon dioxide that were produced by the mitochondria are now what are being taken in. They're now the reactants along with some of the water to make glucose, to store that. We're storing that carbon dioxide as glucose. What they don't include in this particular equation is the fact that light energy is needed also in order to be able to put those 6 carbon molecules together into a glucose molecule. All right, moving on. Next, why can you? Oh, it does say control. I'm sorry. I couldn't see the top of it. I'm being clueless. Okay. So this diagram, please look at this diagram carefully. It's talking about free energy on the side, and it's talking about if we moved directly from combining hydrogen and oxygen, which is what we're going to do at the end of cell respiration, at the very end of cell respiration, we're going to end up combining those to make some water. If we did that in one step, we would have an explosion of heat and light. And that would be very bad to have an explosion of heat and light inside of our bodies. So instead of releasing all of this energy at one time, which we cannot harness most of the energy, and most of the energy would get wasted, our body instead incrementally removes a little bit of the energy, a little bit at a time, and it's releasing just a tiny amount of energy, so not as much heat gets released and not as much energy gets wasted. Here's an example. If you're like, what on earth is she talking about? Let me just give you an example. You have an automobile that you don't maybe personally have an automobile. Imagine an automobile that has a gas tank in it. Now, I could, if I wanted to, drop a match into that gas tank to get your car to move. It would explode, ba-boom, at least I think it would. If it exploded, ba-boom, your car maybe would be flung 10 feet forward, something like that, and we would have wasted a ton of the gasoline to fire, to heat, to light, and we only would have moved your car a few feet. Instead, we have figured out how to build a combustion engine that allows just a tiny amount of gasoline to be run through the spark plugs start it on fire, just a tiny amount at a time, and we release that energy very, very, very gradually instead of all at one time. And when we release the energy that's stored in your gas tank a little bit at a time, it's much more efficient. We don't waste that energy as heat, although our engines do heat up and that is wasted. It's not wasted like an explosion. And we're able then to propel your car however many miles your car can go on one tank of gas. What is that? Like 200 miles? I don't know what the average car can go on their tank. So this is what the body is doing. The body is taking energy. You're going to see this happen. And releasing the energy very, very, very slowly so that it can be as efficient and capture as much of that energy as possible. So it says down here, metabolic reactions are tightly controlled in multi-step metabolic pathways. And that's what we're going to look at today. This is a fun video. If you have time to watch it, you can. They're going to burn a peep and you're going to see a lot of energy being released. Glucose sugar has a ton of energy in it. We may as well watch it for a second. We're wasting time here, but whatever. It's kind of an amusing video to watch. Oops. There we go. So I wish I could fast forward it easily. I don't know if I can. So what all they're doing is heating up a peep. So that's a hot plate that's underneath the peep. And they're just trying to show you how freaking much energy is stored in a stupid little peep. There's a ton of energy. They didn't add anything else. It's just a peep that they're burning here. But peeps have an enormous amount of energy stored in them. That's what a calorie is. A calorie is the amount of energy stored in it. All right, so if I got to figure out how to keep going here. There we go. We're going to move on. So redox reactions. We talked about redox reactions in chemistry class. It's a word where they combine the word reduction and oxidation into one word, and they call them redox reactions. So redox reactions are chemical reactions where electrons are being moved from one atom to another, an ionic reaction. And we've seen that a million times in chemistry class. So take a look at this a little bit closer. Oops, I was hoping this one. There we go. So here, I'm doing everything wrong. There we go. The sodium is becoming oxidized because it lost an electron. Notice that it now has a plus sign because it lost an electron. Chlorine, on the other hand, becomes reduced. And this is always confusing because it gained an electron. The reason it's called reduced is that its charge is lower. It now has a -1 charge because it gained an electron. So that's why we call it reduced. So anything that gains electrons is reduced. Anything that gives up electrons, that's oxidized. And that's going to become, we're going to use those words numerous times as we're coming forward here. This is another example of an oxidation reduction reaction or a redox reaction. In this case, CH4, you might remember, is methane, which is a gas, combined with oxygen in a given environment. This is a combustion reaction. In this case, the CH4 becomes oxidized and the oxygen gets reduced, and it gives off a ton of energy. That's what we're talking about with cell respiration, only we're going to do it in a very controlled manner. It's going to happen very slowly. Couple things you need to know before we can even talk about cell respiration are the electron shuttles. And I don't think that's an official term, but it's a good way to think of that. So we have two molecules in particular. We're going to learn about a third one when we talk photosynthesis. But right now we're going to talk about two molecules. One of them is called NAD+, and the other one is FADH. Or FAD, maybe it's just FAD. Yeah, just FAD here, sorry. FADH2 later. And these molecules have the ability to capture electrons and then carry them somewhere else and then drop those electrons off. So it says instead of direct transfer of electrons, trying to read it better, from glucose to oxygen, instead of going in one step, cells decouple, they separate the metabolic oxidation and reduction. So we're going to separate those two processes and slow them down. The electron shuttles are compounds that store electrons from food. So these are the two that we're going to talk about in this set of notes. So NAD+ and FAD. They exist in both oxidized, that's over here on the left side, and reduced. Remember, reduced means that they've gained some electrons. They've also gained some hydrogens. So it says in cells, electrons come with protons. Now let's practice that again. Remember that hydrogen is just one proton and one electron. That's all it is. If you remove the electron, you have an H plus. We can call that a hydrogen ion, or we can just say what it really is. It's just a proton. So when it says in cells electrons come with protons, they're saying electrons come with H pluses, hydrogen ions. It's a synonym with the word proton. All right, then the transfer of electrons and protons to other areas of the cell use these molecules, use these shuttles to continue metabolism. And you're gonna know all about them in just a minute. All right, what I want you to do right now is to get the big picture of what this Prezi is trying to show you, because it's a little bit subtle. This red box here, this red box that's going around the outside, that represents the entire cell. So notice it's been labeled down at the bottom with the word cytoplasm. Do you see the word cytoplasm down there? Okay, that's outside of the mitochondria, but inside of the cell. This yellow box, that represents the mitochondria. Now, this is completely not to scale. Any given cell might have 1,000, 2,000 mitochondria in it. We've just got one box that's representing one of the mitochondria in it. And then within the yellow box is another box, because remember that the mitochondria has a double membrane. So the yellow represents the outer membrane, the blue represents the inner folded membrane. We call those folds cristae, remember? And then the inside of the mitochondria is called the matrix. The fluid that's on the inside is called the matrix. Now, here's the part where it gets weird, I think. It turns out that the space in between the outer membrane and the inner membrane is wickedly important, and that's called the intermembrane space. And notice they've got all these images of things that are happening in this space in between the membranes. So that's going to become really important. So look at the big picture here. You can see some of the processes that are going on. Glycolysis, glyco refers to sugar. Lysis, remember, means to break. So we're going to break glucose. Glucose is the example we're going to use. Later on, we'll look at other things as well. We're going to break glucose, and that happens out in the cytoplasm. If there's no oxygen present, if you're not breathing well while you're exercising, then you're going to do fermentation, and that lactic acid fermentation is going to happen out in the cytoplasm as well. And that's how you're going to make a tiny amount of ATP. However, if oxygen is present, if you're breathing well, then after glycolysis, the molecules that gets broken up, those are going to get transported into the matrix of the mitochondria, and then all kinds of crazy things are going to happen there. And so you can see kind of the layout of this particular Prezi, and hopefully you'll get the hang of what we're trying to do here. All right, so let's keep moving here. So respiration, cell respiration, to be clear, is catabolic. Catabolic catabolism refers to the breaking down of molecules. Anabolic or anabolism, that's putting molecules together. So cell respiration is about taking glucose, I make it look like a ring. It's about taking glucose and breaking it apart. So that's catabolic. And when you break glucose, remember, glucose is an energy storage molecule. So you're giving off energy. That makes it exergonic. If energy is released, if heat is released, that's an exergonic reaction. So that's what we're doing in this, in cell respiration. It's catabolic and exergonic. And then again, we saw this already. The glucose is what gets oxidized. So that means it's going to start giving up its electrons. And the oxygen is actually going to be at the very end of the whole process. And it's going to capture. I always have students think of the oxygen like a catcher's mitt in a baseball game. And it's going to catch all of the electrons, and imagine like electrons are baseballs or softballs or something like that, and that's what the oxygen is catching. So the oxygen will be reduced, it'll catch some of the hydrogens also, and it will become water in that case. All right, moving on. This is a super helpful diagram, and it would be totally worth your time to pause the video, to stare at this diagram, even at some point when you're studying, to try to draw this diagram from memory. Just be able to understand what's happening, all the different processes. So this is the overview of what's going on. So it says, my picture is kind of in the way at the moment, all respiration begins with glycolysis. So that's pronounced glycolysis, but I say glyco because it's the sugar, glucose, and then we're lysing it, we're breaking it down. So that begins in the cytoplasm. So see right here in this green box, we've got glycolysis, and we're going to break glucose into two molecules that each have 3 carbons. Glucose has 6 carbons. We're going to break it in half, so each of the new molecules has 3 carbons. So pyruvate. Pyruvate gets brought into the mitochondria, inside the mitochondria, into the matrix of the mitochondria. And then it gets slowly, carbons get plucked off of it. And as carbons get plucked off, we start releasing them as carbon dioxide. So that's why, and it doesn't show you that in this particular diagram, but that's why the mitochondria is making carbon dioxide that has to be exhaled out of your body, because it's pulling carbons off of that original glucose molecule that had 6 carbons. So those are going away. As those carbons go away, we're getting energy to make our ATP. In the citric acid cycle, we make a tiny amount of ATP. Oh, I forgot to mention, we make a tiny amount of ATP during glycolysis also. Not enough for a giant organism like us to be able to survive. So it is later on, the electrons get carried by these shuttles. Remember the ones we just learned about? So these shuttles carry the electrons over here, and we have a very controlled set of chemical reactions that happen that release the energy very, very gradually. And then we can produce as many as, I think, maximum is usually 32, maybe 34 ATP total. So just giving you sort of a ballpark of how many ATP we're going to produce. So it says anaerobic respiration. An means without. Air means oxygen. So without oxygen, that's anaerobic. So if you're trying to work out, if you're running and you're not breathing well, or if for some reason you're suffocating, God forbid, if you don't have enough oxygen, if you're experiencing asthma and you're having an asthma attack, and you're not getting enough oxygen to your body, you need that ATP. So your body starts to do something called fermentation. That will also happen in the cytoplasm, not in the mitochondria. The oxygen has to be there to catch the electrons. And if oxygen is not present because you're having trouble breathing or you're not breathing well enough, then we're not going to use the mitochondria. We're just going to do this process of fermentation. So it says aerobic respiration will occur in the mitochondria of eukaryotes. Remember, prokaryotes don't have mitochondria. I'll come back to that in a second. It will involve the citric acid cycle. There it is right there, followed by everybody's least favorite phrase, oxidative phosphorylation. Okay, pause. Oxidation means electrons are given up. To phosphorylate means to add a phosphate, and usually that's for ATP. So you have ADP, adenosine diphosphate, and we add a phosphate to make ATP. So the point of all of this is to make ATP. That's what oxidative phosphorylation is. Now, I just want to make one other quick comment. It says this happens in eukaryotes only. If this, remember that the mitochondria, endosymbiotic theory, remember that they think the mitochondria was once a prokaryote. So prokaryotes can do cell respiration, but they don't have mitochondria. They're the same size as a mitochondria. They do all of these same processes on their inner membrane. So they have folds inside their cells, inside the cell. It's a single-celled organism. They have all these folds, and they do the same processes that we do in our mitochondria, but they do it on their inner membranes. All right, whoops, moving on. Okay, let me get myself out of the way. So here's our big picture. Remember, the red represents the entire cell, the yellow box represents the mitochondria, and the blue box represents the inside of the mitochondria. All right, so the first thing, that we're going to talk about is glycolysis. Now, glycolysis, again, is when we take glucose that has 6 carbons and it's in a ring form, and we break it into 2 molecules that have 3 carbons each. And those molecules are called pyruvate. It takes 2 ATP to break glucose in half, but in the process of glycolysis, we also make 4 ATP. So the net total amount of ATP is a +2. We've actually produced 2 ATP. Now that's good, But that's not enough for a giant multicellular organism like us. We need way more ATP than that. So glycolysis is helpful, not helpful enough. All right, and then the other thing that I will mention, remember our electron shuttles. We had NAD plus and FAD. So during glycolysis, two NAD pluses capture a couple of hydrogens and a couple of electrons, and they are reduced because they gained electrons. That makes them reduced to two NADHs. Now that's going to become super important later on. The NADHs and the FADH2s, they're the ones like our spark plugs in our car. They're the ones that are going to release the energy really gradually so that our car doesn't explode, so that we don't explode with one release of energy and waste all of our energy. So pay attention to the NADHs. Now, this section right here, so much too much information. You guys don't need to know half of this, but still look at the diagram so you feel like you have a clue. So here we are with glucose. We're going to use ATP to break up the glucose. If we travel through, you're going to see that there's all kinds of enzymes involved and all kinds of permutations of the molecules. But as we go, if I go through this entire process, keep moving, keep moving, keep moving, keep moving, keep moving, keep moving, we see that there is a molecule of pyruvate. And how many carbons does it have? 1, 2, 3 carbons. And it says right here, this process happens twice. So there are 6 carbons from the original glucose molecule. Now we've got it into 2 3-carbon molecules that are called pyruvate. All right, so if we jump ahead and we look at sort of the sum total of what's going on with glycose, so what goes in and what goes out, what you need to know is the net. Glucose goes in, and what you're left with are 2 pyruvates and some water, and you don't need to know the water part. Just be aware that you make 2 pyruvate molecules. In the process, 4 ATP are formed, 2 ATP are used, so our net output is 2 ATP. We've made 2 ATP. And then we said this earlier, we've got 2 NAD, oh my gosh, I hate it when it does that, 2 NAD pluses, plus we collect 4 electrons and 4 hydrogens to make the 2 reduced NADHs, and then we have 2 hydrogens left over, 2 hydrogen ions. They'll get used a little bit later. All right, now I mentioned earlier a phrase called oxidative phosphorylation. Whew, that's a very difficult phrase. So oxidative, remember, means that electrons are being given up, and then we're using that energy from the electrons being given up to add a phosphate onto ADP. That's oxidative phosphorylation. A couple of times during cell respiration, there is also something that's called substrate level phosphorylation. So oxidative phosphorylation, you're going to learn about, has to do with the electron transport chain. That's not this. This is directly involving an enzyme. Remember that the substrate for an enzyme is the molecule that it fits with. That's the substrate. This is the enzyme. My claw is the enzyme, and my fist, your fist, is the substrate. Okay, so the substrate in this circumstance has a phosphate attached to it, and then the enzyme attaches the phosphate to the ADP to make ATP. That's called substrate level phosphorylation. It is different than oxidative phosphorylation because there is no electron transport chain. Now, right now, those are words that are going over your head because you're like, I don't know what the heck an electron transport chain is. Just trust me that they're different, and it'll start to make sense as we go on. So let me just read what it says here. Substrate level phosphorylation is a description for when ATP is produced by enzymes transferring phosphate from another organic phosphate. So adding the phosphate onto ATP when an enzyme does that. This is how ATP is produced in glycolysis and the citric acid cycle. So that was in the matrix of the mitochondria. We'll come back to that a little bit. All right. Fun fact, all kinds of dorky facts. Glycolysis is hypothesized to be the most ancient metabolic pathway present in modern organisms, and it happens in all organisms, interestingly enough, and we're going to talk about that more in just a moment. All right, so the inputs and outputs, one more time, of glycolysis. What's going in? Glucose, 2 NAD pluses, and 2 ATP. Remember, it took 2 ATP to get this process started. What's being produced is 2 pyruvates that each have 3 carbons, 2 NADHs, plus 2 H pluses, which they didn't write here, and it bugs me that they didn't write that. Whatever. And then 4 ATP get produced. So that's what's coming out. So the net for ATP is just 2 ATP. All right, moving on. So if this process is aerobic, meaning there is oxygen available, and this is happening in a eukaryote, then we're going to go into the mitochondria. But we're going to pause for just a second. We'll get to that. Instead, we're going to talk about fermentation. That's if it's anaerobic. If there's no oxygen present, if you are having an asthma attack, if you're working out harder than the amount that you're breathing, if whatever, something's interfering with you getting enough oxygen, then you're going to do a process called anaerobic respiration. There are some organisms like bacteria that's all they ever do is anaerobic respiration, fermentation, but they don't need very much ATP, so that produces enough ATP. For you, if you do fermentation, you die. Okay, not immediately, but fairly quickly because you won't make enough ATP to keep your heart beating. All right, so let's talk about anaerobic respiration or fermentation. So give me a second to get out of the way. So NADH, we've got that NADH, it gets oxidized. All of these molecules get recycled, you guys. Your body is an amazing recycler. So NADH loses its electrons and it gets oxidized back into NAD+. So that electron shuttle loses a couple of its electrons. This requires pyruvate to be reduced into another compound, which we're going to call a waste product. So that means that it's going to capture a few electrons, and that's going to change the nature of the pyruvate molecule. This enables glycolysis to keep functioning because now the NAD plus is able to collect 2 more electrons. So it's going to go around and around and around a little cycle. There are hundreds of fermentation pathways. We're going to look at the 2 most famous ones. So the first one is ethanol fermentation. This is where we get our alcoholic beverages from when we make ethanol. This is what yeast does. This is how yeast that you use in your bread makes its ATP. So if you take a look, here's glycolysis. It used up 2 ATP. And then in the process, the whole thing gets stopped because there's no oxygen available. A little bit of carbon dioxide gets produced. A little bit of carbon dioxide gets produced. And we end up with this 2-carbon molecule. We lost one of the carbons. Remember, pyruvate is 3 carbons. We lose 1 carbon to carbon dioxide. So we're left with, who holds their fingers like this? Not me. There we go. We're left with 2 carbons to make our ethanol. And there's our ethanol. So pyruvate, which has 3 carbons, is converted to ethanol, which has 2 carbons and a molecule of and this is screwed up. I don't know why there's a 2 down there, of a 1-carbon carbon dioxide molecule. That's where your carbons have gone. And yeast is what does this process. You do something called lactic acid fermentation. So if there is not oxygen available for this whole process to continue, then we're not going to bother sending the pyruvate into your mitochondria. Instead, we're going to let the NADH drop off their electrons, and pyruvate is going to get converted into something called lactate. This is lactic acid fermentation, and we experience it. You've all experienced lactic acid fermentation. It's what makes our muscles sore and cramp. And it's great that it makes our muscles sore and cramp because it forces us to slow down when we're exercising too much. And then by slowing down, usually if we've been exercising hard, what do? We're like, and we're breathing in tons of oxygen, which then allows us to continue regular aerobic respiration. We can continue making ATP the correct way, and your body can keep its heart beating. If you were to continue in lactic acid fermentation, it's not making enough ATP. It only produces an additional 2 ATP for you. It's not making enough ATP, and eventually your heart would stop. You wouldn't be able to exercise your muscles. So it's a good thing that lactic acid makes us feel cruddy because it forces us to stop and breathe and stop burning so much ATP. All right, moving on. Ethanol fermentation, possibly the most commercially lucrative biological reaction, whatever, making alcohol. Okay, and then the inputs, what's going into fermentation, 2 pyruvates and 2 NADHs, and what's coming out, NAD pluses, and then various carbon waste products like lactic acid, ethanol, carbon dioxide, and there's many other possible waste products. Okay, I'm going to do one more thing and then I'm going to pause this video so that we can split it into 2 sections. So now let's assume that there is oxygen available. So we went through glycolysis, and you can see glycolysis right here. We went through glycolysis. We took we took glucose. We broke it into 2 3-carbon molecules called pyruvate. If oxygen is available, then we're going to transfer that pyruvate into the inside of the mitochondria. So look at our diagram. The yellow is the outer membrane of the mitochondria. The blue is the inner membrane of the mitochondria. So it's going to get transferred to the inside matrix, that's the fluid, of the mitochondria. So let's take a look. Oops. Go. Try that again. And we're going to go inside. So it says, following glycolysis, aerobic respiration of eukaryotes will take place in the mitochondrion. The products of glycolysis are transported through both of the membranes into the mitochondrial matrix. This is where the citric acid cycle occurs. Prokaryotes, remember, don't have a mitochondria because they're the size of a mitochondria. They carry out special aerobic respiration. They utilize special portions of their cell membrane. So they have folds in their cell membrane, and that's what allows that to happen. All right, moving on. Almost done here. So the first thing that's going to happen is the acetyl-CoA. So let's see if we can close up on that. So here's our pyruvate that was made out in the cytoplasm. It gets transported by a transport protein, although the process does not require ATP. It gets transported into the mitochondria, and in the process, 1 carbon dioxide is released. Remember that the mitochondria are making carbon dioxide? Here's one of the carbon dioxide molecules. So that's a carbon. So this was a 3-carbon molecule. We just removed one of the carbons. So we end up with a 2-carbon molecule. We have also added something that's called coenzyme A. So we end up with a molecule that's called acetyl-CoA. They abbreviate it, acetyl-CoA. So that's just 2 carbons. See if there's anything I didn't mention here. CO2 is a waste product that has to be removed. You have to exhale it. Otherwise, it combines with the water of your blood. Remember, CO2 plus water makes carbonic acid. That's bad for your proteins. You can't have acidic or low pH in your blood. That would be bad. The acetyl group is attached to a molecule of coenzyme A. This is the carbon input into the citric acid cycle, which we're going to talk about in a minute, but not yet. Another NAD is reduced to NADH. That means that it has collected some electrons and hydrogens in this case. This is another diagram of it. We're foreshadowing. We're showing you what's about to happen. So this is the pyruvate that was in the cytoplasm. We lose one of the carbons to carbon dioxide. That gets exhaled out. And now we have a 2-carbon, there's the 2 carbons, a 2-carbon molecule that's going to enter the citric acid cycle. Now I'm going to pause here for a second because on the next video, I'm going to show you the citric acid cycle and your jaw's going to drop. It's got like 8 million molecules, 8 million enzymes. It's just so confusing. This is a much nicer diagram because it shows you what's important. You see that there's a 2-carbon molecule that comes in. You see that 2 carbons in the form of 2 molecules of carbon dioxide get released. We collect a whole bunch of electrons in this process. And for every glucose, remember, every glucose makes 2 pyruvates. So that means that the citric acid cycle turns 2 times for every glucose. So we would have made 4 carbon dioxides with 2 turns, and we would have made 6 NADHs with 2 turns, and we would have made 2 FADH2s. So remember that the NADH and the FADH2s, those are the electron shuttles, and they're going to be carrying the electrons to release the energy super, super slowly. So we'll see that in just a minute. And then the last thing I'll mention is one more substrate level phosphorylation. We're just going to make a tiny amount amount of additional ATP during the citric acid cycle. We made 2 during glycolysis. We make 2 during the 2 turns of the citric acid cycle. That's not enough for us big organisms to survive on. All right, and then last thing, we'll just show the inputs and the outputs. Let me get out of the way. So what has gone in is a pyruvate and 2 NAD pluses. What comes out is an acetyl-CoA, acetyl, sorry, you can pronounce it either way. Acetyl-CoA, 1 carbon dioxide and 1 NADH, and that happens twice for every glucose molecule. Okay, I'm going to go ahead and stop our recording here so that you can break this up a little bit.